everybody. Actually, I'd like to present you today the idea that we are developing actually right now in our company. So what we are doing, we are actually dealing with the immune system and with the autoimmunity. So just a few words to introduction. Immune system, if we compare it to the army, is an army that fights with alien bodies that attack our body. And basically within this big army there is a small level of so-called military police. In the immune system, these are so-called T regulatory cells, which basically take care that these immune cells fight with the bacteria and do not fight with the self tissues. In autoimmunity, we've got the situation like this. T regulatory cells, for some reason, do not work, or the level of these cells is too low. So because of that, these cells start to loot. Immune systems start to fight with the self-tissue, start to destroy pancreatic islets, which is the cause of type 1 diabetes, start to destroy our brain, and this is the reason of multiple sclerosis, start to kill our joints, and this is the reason of rheumatoid arthritis. So our idea was to develop somehow these T regulatory cells and provide them to the patients with these diseases, so they can regain control over the immune system, and immune system again get back to the normal and start to fight with the infection and stop to fight with the self tissues. We are mainly focused on type 1 diabetes, and the reason for that is that the type 1 diabetes, the increase in the fresh onset of fresh cases of this disease is continuously increasing, especially in our part of Europe. We are from Poland. So basically right now this is the first chronic disease among children. Type 1 diabetes is mainly children's disease, but this is actually something which happens all around the world. Every year around the world we've got 80,000 cases of new type 1 diabetes, and at least 20,000 of those are eligible for the treatment with our method. So we developed the method of how to first isolate and then expand the T regulatory cells and then give them as a drug to the patients. So what we are doing basically in type 1 diabetes, and this is the scheme of our first trial that is already completed, we're taking blood from the patients with a fresh onset diabetes. We separate T regulatory cells in our GMP facility, expand them to very high numbers, and then give them back to the patient. So this is the autologous product, very safe one. Important is also that in the dialogue with the European Medicinal Agency, we received the classification that this is no longer the transplantation, this is the drug. So we have to do everything according to the GMP, according to the pharma law. So basically, this is so-called advanced therapy medicinal product. And as I said, we performed the first clinical trial, which is already completed. So we've got the first results. It was a dose escalation trial in which we were giving T regulatory cells either in 1 or the 2 doses, starting from 10 millions, then we go to the 20 millions, and then to the 30 millions per kilogram body weight of the patients. And basically, the good story is that we achieved the primary endpoint of the study, so no severe adverse effects of this therapy was found. As you see here, just some minor infections, nothing else. So basically, the therapy is very safe. But of course, apart from safety, we're also looking at the efficacy of this therapy. So whether we can help with the immune system to distinguish between self and non-self and to spare the pancreatic islets that produce insulin in children. I will show you these results of efficacy at the background of the natural course of the disease. So when we've got the diagnosis of type 1 diabetes, patients, of course, start to receive insulin. Initially, the level of this insulin can be at some point in a few weeks at the beginning reduced, so this is the minimum maximum, so some patients could be even insulin independent. This is so-called honeymoon period. But inevitably, within the first year after the diagnosis of the disease, all the patients are on full dose of insulin. All the patients require administration of insulin. At this background, you can see the patients who receive T regulatory cells. So these are our patients. From around 4 months after the diagnosis, we see the first difference in the dose of the insulin which is given to the patient. The patients who receive also the Treg therapy, they require less insulin than non-treated patients. But probably most interesting thing is that even after a year after the diagnosis, we still got some patients after the TREG therapy who does not require insulin at all. Very, very interesting and important results. But probably the most interesting results are here. 2 years and 5 years post-administration of T-regulatory cells, we analyzed the function of pancreatic islets. So what we did so-called mixed meal tolerance test, which is the dynamic test which assesses how many insulin is still produced by our patients, so how many pancreatic islets are still alive. Patients receive control meal, and then every 15 minutes, we measure the secretion of insulin. And as you see here, both 2 years and 5 years after the treatment, our patients who were treated with T-regulatory cells still produce insulin, while those who were controlled do not produce insulin at all. Very important results, because currently children do not die because of the acutes even related to type 1 diabetes. Children die as adults because of the complications, and the complications are related to the not well controlled level of glucose in blood. Neither pen or insulin pump is able to respond very quickly to the changes in the level of glucose in blood, only on secretion of insulin. And these minute-to-minute changes in the internal insulin secretion actually give to these patients control of the glucose level and will delay the complications of type 1 diabetes. So they will probably suffer from this complication not when they are in their 30s, but maybe in the 50s or 60s. So very important improvement in the quality of life for this patient. As I said, this trial is completed, and we've got the observation over the 5 years now, but of course we move to the second trial, which is the phase 2 trial with type 1 diabetes. But before I show you that, I'd like to show you also in what conditions actually T-regulatory cells are currently used around the world. Different types of T-regulatory cells, not necessarily the product that we use. So the first one is a graft versus host disease after bone marrow transplantation, and in fact it was the first trial which started the work with T-regulatory cells, was done in our university, in Medical University of Gdańsk. Actually, we are no longer explore this very routinely. The other field of the use of T-regulatory cells is organ transplantation, and as I said, our company works mainly on autoimmunity. I showed you already the results of the phase 1 trial in type 1 diabetes. Right now, we are conducting phase 2 trial in diabetes and phase 1 in multiple sclerosis. And these are actually the midterm results of these therapies. So first of all, in our phase 2 trial in type 1 diabetes, we modified a bit inclusion criteria. So right now, we include only the patient who still got some level of alive pancreatic islets. So only these patients are included. So we know that they can gain from this therapy. Apart from the group of TREGs and control group, we've got also the arm who received the anti-CD20 therapy to deplete B cells, which sustain the effects of the therapy. It's very important. And these are basically they received 2 doses. In the investigational arms, they received the 2 doses of T-regulatory cells, and the arm with the anti-CD20, they received 4 doses of the anti-CD20 antibodies. And these are the interim results of this study. So control, the red ones, completely lose the secretion of insulin. People who receive only T-regulatory cells are significantly better, but the most sustained effect is in the group of the combined therapy, so T-regulatory cells together with the anti-CD20 antibodies. We will publish the full results of this at the end of the year because we currently in the follow-up. The second study I mentioned is a multiple sclerosis, in which we work with T-regulatory cells. This is the phase 1, so again, we assess mainly the safety. And basically, we give T-regulatory cells either intravenously or intrathecally to omit the blood-brain barrier. We just finished the follow-up, so what I can say, we obtained already the safety results, so there were no adverse effects. Even in this intrathecally injection, we still got very nice results. There were not severe adverse effects of the therapy, and we'll be publishing the full results of this therapy very, very soon. What are the perspectives of the company? We want to further go with the trial with the type 1 diabetes. Right now, we are in the process of scientific advice with the European Medicinal Agency, how to prepare the protocol for the phase 3 trial. So it will be the protocol to register the therapy and to receive the marketing authorization from European Medicinal agency. Of course, we want to continue with the multiple sclerosis in the phase 2 trial. And we are starting preparation for the trial in the rheumatoid arthritis. So these are the clinical points which we are exploring right now. But apart from that, we also developing the next stage product, next generation product, which are the T regulatory cells which are antigen specific. So all I just show you in the clinical trials, these are polyclonal cells. When you inject them, they spread through the whole body. But we found a small population of the cells which are specific only to the pancreatic islets. So right now, because the number of these cells is very small in the peripheral blood, so as you see here, this is really a small promille of the cells in our peripheral blood. Right now we are working on the generation of the good enough number of the cells to be used in the treatment of the patients, and to move it to the GMP conditions so they can be applied as a drug in our further trials, and then we will go to the clinical development of this second generation product. And at the end, just a short story of this discovery. As you see here, we developed everything in the previous century, so we are very ancient. So at the beginning, the therapy was performed at the Medical University of Gdańsk. So first in man administration in GVHD patients happened as academic studies. But then when we started working on type 1 diabetes, our university decided to make a spin-off, POLTREG, to have a vehicle to develop commercially, to develop further the therapy. And as this vehicle, as a POLTREG, we receive significant amount of money to develop the studies which I showed you. And right now, of course, we are looking for the partners to go forward with us with this phase 3 trial in type 2 diabetes, in multiple sclerosis, in rheumatoid arthritis, and also with the development of the second generation product. Thank you very much for your attention.